So a restriction map is used to establish the number and the order of restriction sites, as well as the distance between restriction sites on your cloned DNA segment. The benefit to this is it's going to give you information about the length of your cloned insert and the locations of restriction sites within that clone. So you can determine from the previous whether or not you have a clone of interest based on whether or not it's growing, whether or not it's the correct color. Here, once you isolate the DNA, what you can do is you can determine, yes, my clone was inserted at the correct location and there was no issues. It wasn't, say, inserted backwards. You can do this by cutting the DNA with different restriction enzymes, separating those DNA fragments based on gel electrophoresis, and that's going to separate the fragments based on size. So let's say you have a cloned insert inside of your vector, and you run this on a gel. You cut it typically not at the center of wherever your clone gene is, but on one of the ends. And what this will tell you then is this inserted in the correct orientation. Because, guess what, about 50% of the time, it's actually more like 75% of the time, I don't know why, but math doesn't work out, when you're doing this cloning, it's going to insert itself backwards. And it's not going to be under the control of the promoter if that happens. And so in order to determine that it's in the correct orientation, you can do these different types of restrictions, run it on a gel, and you can make sure that it is indeed in the correct orientation. The smallest fragments are going to move the furthest in the gel. These fragments can then be visualized when you stain it with ruthenium bromide and you illuminate it by this UV light. So here is a sample gel here. To me, this looks like a lane of DNA markers. These are going to be standard sizes so that you can compare your DNA fragments to see if they're the expected sizes or if they're too big or too small. Remember, DNA has a slight negative charge. So when you place samples on a gel and you put an electric charge on it, it's going to be attracted towards the positive end. This down here would be the positive end. And the smaller pieces are just going to be able to move through that gel faster than these larger pieces. You can also see some of these fainter bands here. This is going to be DNA that's in less abundance compared to DNA bands that are very bright. This is going to be DNA that's there in greater abundance. This may come down to how much you loaded on the gel. Maybe this particular sample, which looks fairly faint, maybe you didn't have as much of a sample or as large of a sample as you did with this particular one. So it can mean that, or it can simply mean that when you were doing these cuttings that this piece maybe appeared several times and that's why the band is larger, and so on. So a southern blot is used to identify which of the clones in your library contain a given DNA sequence and to characterize the size of those fragments from Restriction Digest. You can also use it to determine whether or not your clone contains all or part of a gene, to determine the size, sequence organization of a gene, or DNA sequence of interest. And there's going to be two parts to this process. The first part is going to be separation of these DNA fragments based on gel electrophoresis. And the second is going to be transferring that to a membrane and looking to see whether or not radioactive probes will hybridize to that particular region of DNA or not. This is a somewhat busy slide, but it shows how this process works. First, you're going to run your samples on a gel. This is a gel electrophoresis using an agarose gel. This is going to separate out your samples based on size. You then denature your DNA. You don't have to set it up quite like this. There are different ways to set it up, but you basically want to transfer what you have from your gel onto a membrane. And you do this typically using pressure. You may also be able to use, again, a bit of an electric charge to push that DNA from the gel into the membrane. You don't want to run it too long because it can be pushed through the membrane. You want it to just be applied to that membrane. Once it's on a membrane, then you can add your radioactive probe. This is going to bind specifically to your DNA pieces of interest. You wash it off, you wash off the excess. You can then place a piece of X-ray film onto here. That radioactivity is going to emit light. So that light is going to leave little dark spots on your piece of film. So when you look at the piece of film, you develop the piece of film and you look at it, it's going to look mostly clear and light, except for the areas where you had probe bound to your DNA. Northern blot is used, and it's a similar technique, but in this case, rather than looking at the DNA, what you're going to be looking at is RNA, specifically mRNA. And so you use northern blot analysis to determine whether or not a gene is actively being expressed in a given cell or given tissue. And you can use this to study patterns of gene expression in embryonic tissues, in cancer, genetic disorders, or simply look at expression within a particular cell that you would like to study. A Western blot, they're all named after directions. The first one's named after a guy, the Southern, and then they kind of played with it, and then all of these others are variations of the same. So a Western blot is used to look at protein levels in cells or tissue. So again, you're running samples on a gel, except this time you're running protein samples rather than RNA or DNA samples. You separate them out based on size. The smallest proteins are going to run quickest. The largest proteins will remain near the top. A little bit different from the others in that these samples have to be denatured before they're run on a gel. The reason for that is because these proteins may have different shapes, different conformations, and so based on their 3D conformations, they may run a little weird. So in this particular case, all of these proteins are denatured, and so you can know that the smallest, the bands down here are going to be the smallest proteins, the bands up here are going to be the largest proteins. So you then go and you transfer it to a membrane, and you're going to, this time, instead of probing it with radioactive probes, you're going to probe it with an antibody. In this case, they're looking at GAP-DH levels within all of these different tissues. There are going to be thousands of proteins in a cell. You can see here, it just looks like tons and tons of these little blue bands. These are all different proteins. In order to study your specific protein that you want to see, you have to label it with this antibody. And it's a similar process to the others. You're going to wash an antibody over it. You're going to rinse off the excess. In this case, though, you have to add a secondary antibody that will emit light. The antibody by itself, it's not going to look like anything when you put a piece of film on it. So you have to have a secondary antibody to bind to that primary antibody that will glow. You can put a piece of film on it, and then you can see where your protein is and how abundant your protein is going to be. This right here is a technique that I have run probably hundreds, if not thousands, of these gels. It is very, very useful in molecular biology to be able to run these Western blots. You can look to see whether or not your protein is present, whether your protein is being cleaved or processed in any way, and so on. FISH, or fluorescent in situ hybridization, involves hybridizing the probe directly to the chromosome or RNA. So it's going to cut out the intermediate step of that blotting. So in this case, you want to see how is this present, where is it present in vivo. So what you can do, you can carry this out with isolated chromosomes on a slide, you can look at tissue sections, you can look at entire organisms during development. This type of technique is incredibly helpful when you're looking at embryos for various studies in developmental genetics. So for example, here you can see this dark blue color. This is going to be, I'm not sure what gene they were looking at here, but you can see the expression of this, how it changes in these different tissues as this organism is developing. So within DNA sequencing, this is really the ultimate way to characterize DNA structure at your molecular level. And you will often use this in combination with the cloning or with the PCR to get those results that you want to see, whether or not that specific base pairs are the exact sequence that you think that it's going to be. So the most common method, or the cheapest method, is Sanger sequencing. And this primarily was the technique that was used to sequence the human genome. This technique involves the addition of a small amount of modified nucleotides called dideoxynucleotides, and what that happens is it's going to cause the DNA synthesis to terminate. There's a dye attached to this, and it will allow you to be able to read this color termination with a computer sequence. So what's happening here, you have your template strands, you have a primer, you have your DNA polymerase, and you can actually run this in a PCR machine. You have your NTPs, and then you have a small amount of these NTPs, or dideoxy NTPs, that are going to have these fluorochromes. And so each one is going to fluoresce a different color. So in this case, A is going to be a reddish color, pink color, C is going to be a blue, G is going to be a green, and T is going to be a yellow. And so what happens is you do these PCR reactions. Some of them will be short, some will go a little longer, some will go longer yet. And so getting sequence just involves basically running this PCR enough times so that it will cover your area of interest. These are separated out by these capillary gels. This laser will detect the color and it will give you an output for that particular sequence. So this is an example of some of the sequences that you might see as an output, and I have done this myself and you have to go through line by line because sometimes the sequence isn't quite what you expect it to be. Well, I don't know if you can quite see it on here, but some of these early things, instead of giving you a base, it puts an end there. The reason for this is because these peaks somewhat overlap. So when it's doing these early bases here, it's not able to really figure out what is the correct base at that particular position. But as the sequence moves on, you can see these bases are very clear and very distinct, and you can read each of these bases with a particular peak. These other peaks down here are going to be, can mean a couple different things. It can mean that your sample was contaminated with other samples, so that in one sample maybe it read a T at this position, and another sample maybe it read a G at this position, and that might be why you see different peaks. It can also be simply due to error. Once you run this, it only can go a few hundred bases at most, and then you start to run into some errors like this. The next generation sequencing technologies are now being developed, and these are going to allow faster and faster, cheaper genomic sequencing to take place. The drop in price is really, really dramatic. So back when all of these projects were started, it cost about $10 a base in order to do some sequencing. And the total number of sequencing genomes thus was pretty darn low. After these next generation sequencing techniques have been developed, though, you can see this price just drop just really, really dramatically. And right now, it's down to about between $1,000 and $4,000 for the entire human genome. And it's hoped to be brought down even lower, eventually maybe down to $100 for a human genome, so that everybody could have their own genome sequenced if they'd wished, so that they could then see what maybe genetic diseases that they were susceptible for. There are a lot of ethics involved with such a thing. Some people may not want to know their own genetic sequence for various reasons. They'd rather live in bliss that they don't know about it. They may be worried about insurance companies also discriminating against them because they may be predisposed to different diseases, and that's not something to shrug off too lightly. However, there have been laws passed to prohibit discrimination based on someone's genetic composition.